Alright, so we're in our second video here for section 5 of chapter 6. That first video I had you do monohybrid crosses. This video is going to have a dihybrid cross, and also I'm going to talk about some probabilities. So if we dive right in here, a dihybrid cross is any cross involving two different traits. So before we were just keeping track of one thing, the color of the flower. Now we're going to try and keep track of two different traits at the same time and see how they are inherited. Right? As you can imagine, adding extra traits into the mix just makes it a little bit more messy. Right? You have a few more balls to juggle here, but at the end of the day, it's really not that much more difficult than it was before. You just have to keep track of multiple things at the same time, so it gets a little bit harder to do just as far as organization is concerned. So we're going to follow the law of independent assortment here. We're assuming that those allele pairs separate independently during gamete formation. So I'm going to have two different traits. One of them is the color of the peas from those pea plants from before. The other one is the shape of the peas that are resultant in those offspring. So if I'm keeping track of color and shape, I have to assume that those things are inherited independently, meaning they aren't on the same chromosome, so they're going to get mixed on their own randomly in that gamete formation. So Mendel did this experiment, and he ended up observing a 9 to 3 to 3 1. No, excuse me. He ended up observing a 9 to 3 to 3 to 1 phenotypic ratio. And he worked backwards from there to figure out how this whole process works. Luckily for you, I'm going to tell you how the process works, and then you get to understand the law of independent assortment. Believe me, most of us, including myself, don't understand math well enough to be able to work backwards and figure this out. So we're lucky Mendel did it for us. So we're just going to have to dive right in here. These crosses are huge. There's this 16 square Punnett square that you're going to have to use. Again, I'm trying to keep track of two different traits here. The first trait being the color of the pea plant. And Y in this case is going to be the dominant allele for that dominant phenotype. You can have the peas be yellow, or if it's the lowercase y, the recessive phenotype, they could also be green. Then also you have the shape to deal with. If it's the capital R, that's the dominant version for the shape. So those are round and smooth. So it's really smooth, but using a capital S and a lowercase s is kind of difficult, so use an R instead. And the lowercase r is for the recessive trait, and that's a wrinkled pea. Right, so the surface is kind of wrinkly. So those are the two traits, the color and the shape of the pea that we're trying to follow through this. And this is a hybrid cross. So my two parents are heterozygous for both traits. Right, there's one dominant and one recessive for both the color and for the shape. Now the first thing that we have to deal with here in order to do this is we have to figure out what possible gametes those parents can have. So before I can do the big Punnett square, I'm actually going to have to do a smaller one. So I'm going to take one of those parents and their genotype as I can see it, and I'm going to try and figure out what different possibilities there are within that. That's probably not going to be big enough. So I'm going to cross my Y's versus my R's and come up with these different possible gametes. And both parents are going to do that, but then those gametes are going to contribute to your new offspring. So on each side of this Punnett square, I'm actually going to have to use those four different possibilities, those four different combinations. You can have a capital Y with a big R, a capital Y with a little r. You can have a lowercase y with a capital R, and two lowercase over here. Right? And that's going to be the same for both parents. So I'll fill in this side just the same. Okay, so again, this is a little bit more complicated than it was before, but it's really not that different. I'm still going to fill in from the top and from the side. So from the top, I'm going to get a capital Y and a capital R for each offspring in this row. But then I'm also going to fill from the side and find out that this first one is going to be homozygous for both traits, and continue to fill all the way across. Now, what I'm doing otherwise is just a preference, right? I like to put Y's together, R's together, and I also like to put a capital Y before I put, or a capital letter before I use a lowercase. So I'm going to fill this whole thing out, and you should too. But at the end of the day, you really shouldn't be copying me, because it's going to take you longer to get it figured out and get it done if you copy rather than just filling in yourself. Because you should be able to figure it out at the end of the day, from top and from the side, what combination each one of these is going to have. All right, so I got that all filled out. Now all I have to do is interpret it. Again, might take you a little bit longer to get it figured out the first time, but as long as you get the right combination from both parents, right, you should be able to fill these kinds of Punnett squares out appropriately. This next part is actually the hardest part. That's not even that hard either. So I'm going to go through and I'm going to figure out for each individual offspring, for each square in that Punnett square, what traits am I going to see? Right, so I'm going to go for each thing just one at a time. I'm going to start with the color, but I'm just going to look for my yellow offspring. So I'm going to go through and I'm going to highlight anything that has a capital Y, because that would automatically make that a yellow P. So anything with a capital Y needs to be highlighted. So that covers all of them. Anything that's left is probably green, because I have that homozygous recessive. Two lowercase y's gives me that recessive trait. Those ones are going to be green. And then I go through and I try and figure out how many of those offspring are going to be round and smooth versus wrinkled. So I'm going to use orange for smooth, and all I have to do again is go through and look for anything with a capital R, because that should show the dominant trait. So I see capital R's for all of these offspring, and then the other remaining ones obviously are going to be wrinkled. They have two lowercase r's. So I got those highlighted. Right, now at the end of the day, I want to know what my phenotypic ratios are. How many of the offspring are yellow and smooth. How many of them are yellow and wrinkled? How many of them are green and smooth? How many of them are green and wrinkled? So there's 4 different possibilities. Right? You have 2 different colors, 2 different shapes. You just have to come up with those ratios for each one. So I'm going to go through and I'm first going to count how many of them are going to be yellow, so they have at least one capital Y, and smooth, so they have at least one capital R. So I'm going to go through and look at that. So it's got to be yellow and orange. So I see 1, 2, 3, 4, 5, 6, 7, 8, 9 different squares that have that combination. So for yellow and smooth, there are 9 offspring that have that combination. Alternately, they could be green and smooth. So I got to count those up. There are 1, 2, 3 of those. In my bottom corner, down on the right-hand side, I have 1, 2, 3 that are going to be green and smooth. Right? I probably should be crossing these things out as I go, or at least checking them off. These were my yellow and smooth, just so you guys can follow along and see what I'm doing. But next thing that I want to look for, instead of yellow and smooth, how many of them are yellow and wrinkled? And if I go through and look at that, there are 1, 2, 3 of those. And finally, the last possibility, they can be green and wrinkled. So I would have to have 2 lowercase y's and 2 lowercase r's, and there should just be 1 of those, this one left down at the bottom corner. And as you can see, there's a 9-3 to 3-1 ratio. So what Mendel did was he observed 9-3 to 3-1 ratios in actual populations of these flowers, just counting how many of them had yellow and smooth peas and how many of them were green and wrinkled, how many were green and smooth, how many were yellow and wrinkled. And he came up with this data that gave him that 9-3 to 3-1 ratio, and he worked backwards to find out that this is how it works. So again, lucky for you, you don't have to worry about it that way, but you are going to have to do a few of these Punnett squares and interpret them. So hopefully, at least working off of this example, you can start to do that.